So I would like to address mainly 3 points in my presentation, give a brief overview of the Genome-Wide Association studies, present our own results of this approach for adiposity-related traits in the Quebec family study, and address some of the challenges in the study of gene-environment interaction in the era of Genome-Wide Association studies. So if we look at the slide that presents how we went from candidate gene studies to Genome-Wide Association study. So in the whole time we used to, in association studies with candidate genes, test whether a gene and a trait were associated within a population. This is the basic goal of association studies. And in candidate gene studies, genes obviously were selected based on prior knowledge about the possible function of the gene and in the etiology of the disease, or on the basis of their location within a QTL, what we call positional candidate gene. So in a genome-wide association study, basically it's an association study performed on the whole genome. And in that strategy, the SNPs or the variants are selected based on the basis of the linkage disequilibrium pattern in the genome. And in fact, if we look at that figure, if we assume that this is the causal variant, we type a lot of SNPs around a gene or in the whole genome on the basis of linkage disequilibrium. We can capture most of the variation, and if we find an association with a given SNP, it is probably in linkage disequilibrium with the causal variant, which means that when we find an association in a genome-wide association study, basically we find a locus, not necessarily the causal variant. So genome-wide association study use a large cohort of cases and controls. It can also be done on family studies, and we use the technology of the microarray-based SNP genotyping, in which we have on an array several thousands and even millions of random markers, and we try to detect association with each of these markers with the disease or a quantitative risk factor. So it has been this approach has indeed been very successful, and there's a catalog of the GWAS results published on this website, the National Genome Research Institute. And as you see, as June of 2009, there were 439 published GWAS results. And 1 year after, June 2010, 904 published GWAS for 165 traits. So in just 1 year, we are more than double the results emerging from GWAS study. And as you can see, signals have been found all over the genome for either disease or quantitative traits. This slide presents the relationship, the inverse relationships that we can see between the allele frequency in a variant and the effect size of the variant. And as you can see, obviously, the relationship is inverse. The more common is the variant. Like here, this is typically what we are looking at in genome-wide association study. Generally, the effect size is very low. And this is basically what we are targeting in genome-wide association study. And if we try to look for low-frequency variants or even more rare variants, which effect size are greater, they are not typically looked after in the genome-wide association study. So we are talking in genome-wide association study of variants with a frequency, minor allele frequency around 5%, while low-frequency variant would be between 0.5% and 5%. So there are, I think, 3 major characteristics of the loci that have been identified so far through genome-wide association study. First, it is important to remember that this approach is based on what we call the common disease, common variant hypothesis, which states that the genetic susceptibility to common disease is attributable to a limited number of variants present in more than 1 to 5% of the population. As I said, typically, these variants have a small effect size with odds ratio ranging from about 1.05 to 1.3. They generally explain a very small portion of the heritability of the disease or the trait. And so far, the trait-associated SNP that has been located using this approach are found in outside genes. 30% of the SNPs association have been found in intergenic region, 40% in non-coding region, and about 20% in protein coding regions or in tight linkages of equilibrium with those protein coding regions. And we also discover, using this approach, a lot of unsuspected association of genes with certain conditions. I think in the field of obesity, a good example is certainly the strong association that has been found with the FTO gene, which encode a protein involved in the demethylase. Just to give you an example of the type of results that we can get from Genome-Wide Association study. This is a recent study published in Nature Genetics in October, in which they look for association with body mass index, the indicator that is used in most of these studies to index obesity. And as you can see from these results, they were able to find 32 loci associated with BMI. What you have in this slide is those represented in blue were the previously identified loci from GWAS. And with that study, using a very large number of individuals, they were able to find 22 more loci. 4 of them, indicated in green, were previously found to be associated with body weight or waist circumference. And they were able to uncover 18 new loci. And as you can see, obviously, FTO stands out here with a very impressive value of 10 to the minus 60. But the point here is that these 32 BMI loci, the variance accounted for by each of these locus for BMI range from 0.01% to 0.34%. And overall, these 32 loci explain just 1.45% of the variance in body mass index. So that's led the researcher to argue that there are missing irritability and try to understand what could explain this missing irritability. A few factors could explain this missing irritability. The first one is that there are probably other types of variants that are those that we are looking at in Genome-Wide Association study. Basically low frequency variants or rare variants, structural variants like copy number variants, of course factors like epigenetics, epistasis, gene-environment interaction, and even some authors argue that there is no missing irritability because the irritability refers to the additive genetic variants, what we call the narrow sense irritability. But most of the time when we calculate irritability for disease or quantitative traits, we are not specifically calculate the narrow sense irritability. So if we just look at the additive genetic variants, there might be no missing irritability. Or another very likely explanation is that there is many more common variants with too small an effect size to reach significance in Genome-Wide Association study. And just to illustrate that last point, I would like to show you the results of a recent study in which they look at a common SNP that could explain a very large proportion of the irritability of human height. As you know, height is a highly irritable trait, and irritability around 80%. And recent Genome-Wide Association study dealing with that trait have identified 50 common polymorphisms in that trait, accounting for about 5% of the variance. So what the authors did in that study is they estimated the proportion of variance for height explained by close to 300,000 SNPs, genotype on close to 4,000 individuals. So basically what they did here is they use a classical quantitative genetic approach by regressing height on genetic relatedness of individual inferred by the genome-WAS genotype. So basically this regression coefficient is equivalent to an irritability, and in that case the irritability represents the proportion of variance explained by the SNP. So they were able to find that 45% of the variance in height can be explained by all these SNPs. So it's a big increase from the 5% of the variance explained by the GWAS previously published. And after correcting for incomplete linkage disequilibrium between the tag SNP and the causal variance, the explained variance reached 54%. And even if we account by modeling the causal SNP with a minor allele frequency of less than 0.1, the variance explained reached up to 84%. So these results suggest that most of the irritability is not missing, but is rather hidden because the effects of the SNPs are too small to be significant. Of course, we don't know if this applies to common disease or quantitative risk factor traits. But if it's the case, that means that probably we need to look for more variants. So what can be done to make the most of existing and future GWAS? I think obviously we need to ensure wide availability of the data to increase sample size and ensure thorough meta-analysis. We need to mine the existing GWAS for association with structural variants and evidence of gene-gene and gene-environment interaction. And one of the arguments that has been put forward in that review was to focus on the discovery efforts on well-phenotyped populations in order to reduce heterogeneity and explore pleiotropic effect. And indeed, we can also use family studies in that context. Family studies has some advantages. They facilitate the detection of rare and low frequency variants because they tend to co-segregate in families, facilitate the investigation of gene-gene interaction, permit the investigation of parent of origin specific effect. We have evidence that for some disease like type 1 diabetes and type 2 diabetes, the effect depends on where the allele is originating from one parent or the other, and also exploit the information from linkage studies to prioritize SNPs. So having family studies, we can use linkage and use that information to be able to prioritize the SNPs in association study. So we use this approach in our Quebec family study, and this was made possible as an effort of a team grant led by Dr. André Marette in Quebec. And the team grant purpose was to study the cardiovascular complications in type 2 diabetes. So this was done in collaboration with a different group of people in Quebec, and also with my collaborator in San Antonio from the Southwest Foundation for Biomedical Research, in which the genotyping was done for that study. So the main objective of this genetic core of this team grant was to identify genes involved in the development of cardiovascular complication in type 2 diabetes. Our main hypothesis being that inflammation plays a key role in the pathogenesis of obesity-linked disease. So genetic variants across the genome are associated with metabolic and inflammatory pathways linking increased body fat accumulation to type 2 diabetes and cardiovascular complication. So we take advantage of the well-phenotype data that we have in the Quebec family study to perform a genome-wide association study. So we had DNA available on 928 subjects. The average BMI was 27.5, and the phenotypes investigated that I will present today are waist circumference, the abdominal adipose tissue areas measured by CT scan in the Quebec family study, and some cytokines, CRP, adiponectin, IL-6, and TNF-alpha. So we use the Illumina 610 quad chip, which contain more than 600,000 markers, with a median marker spacing of 2.7 kilobases, and this chip captures about 93% of the variation in the whole genome. After the genotype quality control processes, the average call rate was 99.7%. We exclude SNPs with call rates less than 95%, monomorphic SNPs obviously, and those with a minor allele frequency of less than 1% and not in R.D. Weinberg equilibrium, which led us to a total of 543,714 SNPs that were retained for analysis. And we test association with the measure genotype approach implemented in the Salud software. And all the data that I will present, the phenotypes were adjusted for age and sex. So here I show the results for waist circumference. So you see here the results for each chromosome. So each of the dots that you see there represents an association with a given SNP. And as you can see for waist circumference, the best signal was obtained on chromosome 6 in a gene called NOX3. And the p-value was 3.1, 10 to the minus 7. And in fact, this particular SNP was not directly in the NOX3 gene, but was about 400 kb from the NOX3 gene. And the variance in the waist circumference accounted for by this particular SNP was 3.5%. This table presents the best hits that we had for adiposity-related traits in our cohort. So for BMI, you see that the best hit was with a gene called serine C5. For person body fat measured by underwater weighing, the best hit was with a gene called CDH13. For waist circumference, as I just said, was with NOX3. And for total abdominal adipose tissue, the best hit was also with CDH13. And as you can see, for all these adiposity traits, NOX3, we found evidence of association with NOX3 and with the particular SNP that you see on that slide. This slide presents the best hit for the cytokines. I won't go over them in details, but you can see that for adiponectin, we found the best signal with a SNP in the adiponectin gene with a p-value of 2.5, 10 to the minus 9. And that was the best hit of all the traits that we analyzed in our GWAS. So we were interested to see if there were some common variants that could explain adiposity and the cytokines. And as you can see in that table, indeed some of them were associated with adiposity and to a lesser extent to the cytokine variables. This is the case for NOX3 and CDH13. And we think that these two genes represent probably good candidate genes linking adiposity, inflammation, and insulin resistance. NOX3 is a gene of the coding for a member of the NOX family of NADPH oxidases. These are enzymes having the capacity to transfer electrons across the plasma membrane and to generate in that process reactive oxygen species. ROS production is enhanced in the adipose tissue of obese animal, and this NOX-enhanced generation of ROS promotes the production of TNS-alpha. ROS also enhances adipocyte differentiation. CDH13 is the cadherin 13 art cadherin T protein, which encodes a calcium-dependent cell-cell adhesion glycoprotein, which is predominantly expressed in vascular endothelial cells and bind with specific isoforms of adiponectin at vascular, endothelial, and smooth muscle cells. It is upregulated in vascular endothelial cells within atherosclerotic lesions, and it has been associated in previous GWAS study with coronary heart disease, blood pressure, and adiponectin levels. There's at least 4 or 5 GWAS studies that have shown that this particular gene was associated with adiponectin levels, I think including 2 studies in 2010. So taking advantage of our collaboration with our Finnish partner in that team grant, we take advantage of a Genome-Wide Association study undertaken in a Finnish cohort called the UGene2, and also try to replicate some of the results in a sample called the METSIM study, the metabolic syndrome in men study, which include more than 10,000 individuals. So in the UGene2 Genome-Wide Association study, data were available on 900 subjects with a total number of SNPs over 500,000. And we look at several SNPs in our 2 candidate gene study in that cohort. And for both NOX3 and CDH13, we find evidence of association with different SNPs, although they were not the same SNP that we found in the Quebec family study, for BMI, person body fat, waist circumference, and also with CDH13 with person body fat and visceral adipose tissue with some pretty good p-value for the visceral adipose tissue. We specifically look at the effect of the SNP that was associated with waist circumference in the Quebec family study. The same SNP was also available in the Heritage family study. And what you see in that slide is the impact of that SNP on BMI, waist circumference, person body fat, and total abdominal adipose tissue, which has been measured the same way in the Heritage and the Quebec family study. And as you can see, the variant here is associated with increased adiposity in both cohorts. So that is true for BMI, waist circumference, person body fat, and total abdominal adipose tissue. So I think we can see that NOX3 certainly appears to be a strong candidate gene linking adiposity, inflammation, and insulin resistance. And I think we have 2 lines of evidence suggesting that. The first one is, as you know, elevated free fatty acid levels has been associated with obesity, and this is thought to play a pathological role in the development of insulin resistance. A recent study from Dr. Maris' group was able to show that circulating lipids inhibit hepatic insulin action, and this inhibition is made via the induction of INOS, which leads to an increased production of nitric oxide, probably to the production of an increased peroxynitrite. This has been published in Diabetes in 2010, and Andre's group has developed a knockout mice from this INOS. And when we look at this effect in the knockout mice, they were completely protected from this lipid-induced hepatic and peripheral insulin resistance. The second line of evidence comes from a paper that was published in July, looking at the relationship between oxidative stress, which has also been implicated in the pathogenesis of insulin resistance. And in that study, the authors showed that palmitate-induced hepatic insulin resistance was mediated by NOx3-derived reactive oxygen species, which was shown by an upregulation of that specific gene in hepatocytes and not an upregulation of the other members of the NOx families. And they were able to show that NOx3 downregulation led to a reduced ROS generation and improved insulin resistance. So it appears that these enzymes, which produce the superanion oxide, and you have here the INOS, which produce nitric oxide. The superanion reacts with the nitric oxide to produce the peroxynitrite, which has evidence that it could impair insulin action. So I think the link between NOx3 and this INOS certainly is a strong indication that NOx3 could be a good candidate gene linking inflammation, insulin resistance. Now the last point I would like to address is how can we deal with the genome-environment interaction in genome-wide association studies. This slide just presents you a trend in the human genome epidemiology papers that were published in the last 10 years. I took this data from the Huge Navigator website, in which I will try to compare the number of papers that were from GWAS studies versus the gene-environment interaction studies. And as you can see, in 10 years, obviously we had a lot of increase in papers dealing with basically association with disease or quantitative traits. Obviously, in the last years, practically the last 2 years, a very strong increase in the papers dealing with genome-wide association study, but which represent about from 1 to 5% of the total paper. But the last column presents the percentage of all these papers dealing with gene-environment interaction. And as you can see, the percentage has been quite stable over the years, which means that so far we certainly had not took advantage of the GWAS data to exploit gene-environment interaction. So what are the challenges in the study of gene-environment interaction in the era of genome-wide association study? I think the major one is certainly exposure assessment. It is very difficult to obtain accurate and reliable measure of the environment. If we think of obesity, diet and physical activity are those main environment factors. And of course, exposure may vary over lifetime. So we are desperately in need to have good biomarkers of long-term exposure. Another very important issue, of course, is the sample size and power. To detect gene-environment interaction in genome-wide association study, we need very large sample size. And a rule of thumb said that we need at least 4 times larger sample size than the one required to detect a main effect of comparable magnitude. So that means that in a GWAS design, we would need tens of thousands and hundreds of thousands of individuals to detect gene-environment interaction. And the last issue is certainly the heterogeneity, in terms that across the different studies around the world in which we have access to DNA, different tools have been used to assess exposure. And because of that, just this single issue makes replication even more difficult. So we need to have access to some consortium to address this issue, like the Geneva Consortium, for which the goal is to identify genetic variants related to common disease, identify difference in gene trait association related to environmental exposure, and of course ensure rapid sharing of the data. So how can we address gene-environment interaction in the context of GWAS? I think we need to investigate interaction with loci that has already been associated with some trait or disease in genome-wide association study by modeling the environmental effects. So I will give you some example of this. First, by showing that with our NOX3 SNP in the Quebec family study, we tested to try to find some interaction with several SNPs in those in this gene and the other candidate genes that I showed you earlier. And in the case of that particular SNP, we found evidence of interaction with caloric intake expressed per kg of body weight. The p-value for interaction was 0.04, but as you can see, the effect of that particular SNP was significant only in subjects with a high caloric intake per kg of body weight, with a value of greater than 32, which correspond to a caloric intake of about 2,200 calories. So you can see that the SNP effect was highly significant in that high intake group compared to the low intake group. Another good example of how we can deal with interaction in the Genome-Wide Association study is a very interesting study published last August in PLOS Medicine, in which they look at interaction with physical activity, based on a large number of subjects, and as you can see. And basically what they did in that study is that they took 12 SNPs in 10 different obesity loci identified through GWAS, and they calculated a genetic predisposition score based on the sum of BMI increasing allele for each of these SNPs. And they regressed that genetic predisposition score on BMI. And as you can see here, on the overall group, there was a significant association, and the regression coefficient was, the beta regression coefficient was 0.15. But as you can see, the effect of the genetic predisposition score was much more important in the inactive group, with a beta regression coefficient of 0.2, which translate in terms of body weight for a person of 1.70 meter tall, in a difference of body weight of 592 grams. So as you can see, the impact is much stronger in the inactive people. And in fact, when we look at the regression coefficient in those two groups, you can see that the regression is higher in the inactive group compared to the inactive group. And you have the difference in terms of body weight with those two groups. And in that case, the variance accounted for was 1.2% here and 0.6%. So if we take the difference in terms of body weight between those two groups, this translates into a 40% difference, which means that an active lifestyle is associated with a 40% reduction in the genetic predisposition to obesity. So even if you have a genetic predisposition, being active is something that could be good. Another way to deal with gene-environment interaction is to model environmental effect in genome-wide association study. And a very interesting study did that by performing a GWAS on total cholesterol, adjusting in two different ways. Age and sex adjusted data, which are represented in blue here in the figure, and another model in which they adjusted for age, sex, diet, and physical activity. And as you can see, in some cases, the adjustment increased the significance of the p-value for a number of SNPs. And in fact, they were able to discover with that approach two SNP, one in the GLUT2 transporter, another one in the HP gene, that stands out after adjustment for this lifestyle. Just to finish, a very last study that was published in Diabetes Care online, that look at the interaction of dietary whole grain intake with fasting glucose and insulin in a very large meta-analysis of 14 cohorts. They studied the interaction with 16 loci associated with fasting glucose and 2 loci associated with insulin concentration. And they found, despite the large number of subjects, no evidence of interaction after adjustment for the multiple testing, which in that case correspond to a threshold of p less than 0.0028. The strongest interaction was found for fasting insulin, only one interaction with a particular SNP in the gene glucokinase regulatory protein. In that case, the greater whole grain intake was associated with a smaller reduction of fasting insulin in subjects carrying the insulin-raising C allele. So to summarize, I think we can say that GWAS are based on the premise that genetic susceptibility to common disease is explained by detectable genetic variants. And as I showed, most of the variants identified so far from GWAS have very small effect size and explain a small proportion of the irritability. Several factors could explain the missing irritability, but I think certainly the presence of gene-environment interaction can weaken or mask a true association. So if we don't take that into account, we can certainly miss something, and I think we can say that in the GWAS world, absence of evidence does not necessarily imply evidence of absence. So GWAS also have tended to focus exclusively on statistical evidence, but I think studies undertaken on well-phenotyped population, which is very often the case in family studies, with proper assessment of exposure can lead to the identification of novel susceptibility loci. The more than 100 GWAS that has been published so far, I think, have provided valuable insight into the genetic architecture of several diseases, and I think attempts should be made to fully exploit the existing data by trying to attempt a functional characterization of the trait-associated loci, mining existing GWAS for association with other type of variants, and of course modeling of environmental factors to investigate gene-environment interaction. I would like to finish and take the opportunity to remind you that the Society has a journal, as we told at the beginning of the session, the Journal of Nutrigenetics and Nutrigenomics. And if you are here, I assume that this is because you are doing research related to that field. So I would like to invite you to submit your contribution to the journal. Thank you very much.